Welcome everyone. This is the first virtual seminar of the Imperial College Physics of Life Network. So maybe, let's go through a bit of the logistics first, right? So we will be recording this session. So just to let the audience know. And during the talk, we invite the audience to post questions on the chat. And if you don't want your name to be shown, then don't put your name down next to your question. And but maybe we can start by asking the director of the network, Professor Robert Andres, here to say a couple of words about the network.
Okay, all right. Thank you very much for being here. Welcome, everyone. I'm, as Stefan said, I'm Robert Andres. I'm one of the directors of this Physics of Life Network of Excellence at Imperial College. Stefan and Chris Dunsby are the other two co-directors. And before we maybe start with the exciting event today, just a couple of words about the network. So the aim was to integrate the different biological physics expertises we have across campus in various departments, bioengineering, math, life science, where I'm located, for instance, into this umbrella. And we had already a few very nice events. We had a launch event a year ago, roughly. This summer, we had a virtual symposium, which was also really exciting, mostly people from London. And then we have been also had this seminar series, not seminar series, a journal club for a couple of years, and that's essentially how it started, this weekly journal club. So and so I think today we are here because of this brand new seminar series, which is virtual for now. And Tom Aldridge and Chu Fan have initiated this, and I guess I'm handing back to Chu Fan and welcome again and so on. I'm really excited.
Okay, well, thank you very much. So let me continue with the logistics a little bit. So like I said, we encourage the audience participants to post questions on the chat, and you can also put put thumbs up if you like the questions, and then we will periodically interrupt Margaret during the talk to ask those questions if there are any. So we would like to encourage the physics tradition of having questions throughout the talk. And I think that would make it more interactive as well. And so that's everything pretty much. So now I would like to really introduce our speaker today, Professor Margaret Gardel from the University of Chicago. And it's my, a great pleasure to introduce her. I'm a long admirer of her work. And so a bit about Margaret. So she did her undergraduate studies in physics and mathematics at Brown University. And then she did a PhD at Harvard and postdoc at MIT, and became a faculty member at the University of Chicago in 2007. And she is currently the Horace Horton Professor in the Department of Physics in Chicago. And quite a few awards, so I will just mention a couple. She was awarded a Sloan Research Fellowship and also received a Director's Pioneer Award from the National Institute of Health. And yes, so maybe I'll just pass over to Margaret. We very much look forward to your talk.
Okay, thank you very much for having me. And I want to encourage the audience, all these black boxes, to please, please, please ask questions, or else it'll feel like I'm speaking into the abyss for the next 40 minutes. So I really encourage you, and I want to make sure that this is useful and interesting to you. And thank you very much for this invitation. As I was saying before, I have young kids at home, so I've made the decision not to travel very much, especially not internationally, over the next 5 years. And so despite all the ongoing worldwide troubles, this is really a bright spot of mine to be able to interact with colleagues around the world. So I'm going to tell you two stories. I think I gave a talk that was more general. I'm going to tell you two stories that just came to completion in my lab. One is about the biophysics of morphogenetic ratchet that controls epithelial cell shape. And then the other one is a new mechanism of force sensing that we found in the actin cytoskeleton. So over the past 5 years, I run a biophysics lab that's interested in how mechanics of the actin cytoskeleton is regulated. So we use in vitro reconstitution to reconstitute actin assemblies outside of the cell, but we also study regulation of cell shape and adhesion. And in the past several years, we've become more interested in the context of epithelial tissue. So these are two of my favorite movies, one from Matthew Tiska's lab at Vanderbilt, where he is imaging fluorescently labeled actin in an intestinal organoid. And this movie is over the course of about 12 hours, and the colors just reflect the different heights of the cell. So it's a three-dimensional spherical shape. So you're able to see the changes in height as the cells are changing shape and undergoing cell division. But you should also see the sort of nice fluid-like rearrangements of the cell shapes as the organoid is proliferating and growing. On the right is a lattice light sheet microscope from Eric Betzik, really showing more detail of the actin filaments that are driving morphogenesis of a developing Drosophila embryo during dorsal closure. And so here you can see this pulsatile nature of the actin networks as the embryo is changing its shape. So, so if we think about the mechanics of epithelial sheets, a model that has been used fairly successfully really harkens back to one that was proposed by Sir Darcy Thompson over 100 years ago in his wonderful book on growth and form. Anybody who is interested in morphogenesis at the cell and tissue scale should really take a look at this book. It's gorgeous. And so what he postulated was that if you look at cells packed in epithelial tissue, then maybe we could consider, it looks like the boundary stresses between the cells are really what's dominating the cell shape. So he postulated that maybe we could take the physics of soap bubbles, of a packed collection of soap bubbles, to really to reflect all these edge tensions and model what the distribution of cell shapes are in epithelial tissues. And so in the basic Vertex model, what you do is model the edge of each cell as having an inherent tension, a force to it, and it has a certain length. And then you additively add all these edge tensions together to get a energy term. And you also add to that a compressible, an area compressibility term. So with that, you get the total energy of the system. And then you can calculate what the predicted distribution of cell shapes would be. This Vertex model that is completely elastic, right? So if you just think of it as a spring, if you change the force of any of the edges, the length will reduce by a certain amount, just like in a traditional spring. And if you reduce that force, it should go back to its original length, right? So it's an elastic picture. It doesn't allow for the dynamic rearrangements that is necessary in living systems. And so this Vertex model has been used extensively over the past 100 years, really, to really make predictions of distributions of shapes in epithelium. And one thing that's become clear, especially as we've been able to test these models more, is these coarse-grained models at the scale of individual cells can work surprisingly well, right? But where I see the field as really limited right now is we don't understand the regulation of the parameters. If we go back to like our epithelial tissue, remember, if you could actually arbitrarily change the tensions of any one of these edges. And in fact, in some pictures that people use, they do. They just sort of arbitrarily change the tensions of the cells, and we don't know how those edge tensions are regulations. So how is the spatial variation? But also, we don't know how any one of those edge tensions evolve over time. So we have, even though we have coarse-grained models where we might have a very few number of parameters, the living systems, there's enough spatial variation and time dependence of those parameters that it makes it really evasive to predictive theories, all right? And so that was one thing that we're trying to deal with, is how do you make measurements on systems where there's dynamically changing parameters with spatial variation? And if you take a step back, when you have materials where their parameters are changing in this way, what this means is there's constant stress rearrangements in the system, and the materials are adapting. The reason they're changing their parameters is they're responding to external cues and they're modifying their behavior, and then that's what allows for morphogenesis. So if we even just think about the fundamentals of the materials that are underlying these types of morphogenic changes, what you need is some way of generating stresses within the cytoskeleton, but also some feedback to sensing forces in the cytoskeleton to be able to construct all these active and adaptive properties. And so that's what I'm going to talk about today, is one way of getting a morphogenetic behavior at the scale of a single cell-cell junction. And so I'm going to talk about the process of this ratcheting behavior and how we could constrain a model of how that ratchet works. And then I'm going to talk about something a little bit more biological, with which we haven't done as much modeling yet, which is a new molecular mechanism of force sensing of stresses in the actin cytoskeleton. Okay. And again, please ask any questions. Feel free to interrupt me. Okay, so I introduced the Vertex model. I'm going to go, for those of you that aren't obsessed with the actin cytoskeleton like I am, I'm going to introduce what the important parameters to think about are in the epithelium. So here is again a fluorescence image of epithelial tissue that we formed with tissue culture cells on a soft collagen gel, and we let them evolve and get packed over over several days, and we image the apical surface of these cells using confocal microscopy. And what we're looking at here, this is a stain for actin filaments. And the edge is high intensity because there is an apical actin belt around each of these cells. That's really determining the shape of those cells. Below, in interactions with the collagen matrix, there's other basal actin network that's coupled to the collagen mesh, and so that's the basal surface that's coupled. And what we found actually, that these two apical and basal interactions, at least in the tissue culture cells, are really decoupled. They're mechanically not interacting with each other as much as you might expect. So what we're going to be focusing on for the next 20 minutes is this actin belt at one of the junctions. This is again another stain of the adhesion molecules that's coupling two cells, that's forming attractive interactions. So there's E-cadherin molecules bridging the two cells. There's actin filaments that are there, and there's also molecular motors of myosin 2 that's generating contractile tension and forces along this junction. So we're going to be asking how this array of actin and myosin and adhesions work together to control the length of this cell-cell junction. Okay, so this actin, we see this actin is assembled at this interface. And the way the actin and the myosin is actually assembled at this interface is controlled through a RhoA GTPase cycle of RhoA. So at the plasma membrane of the cells, there are factors that turn on and turn off Rho signaling. And when Rho signaling is turned on, RhoA GTP stimulates the polymerization of actin through formin, and stimulates the assembly of myosin 2 through Rho-associated kinase. And so it's by regulation of turning on and off this Rho signaling that these actomyosin arrays, these contractile stresses at the cell-cell junctions, are regulated. And so in addition to this mechanical stresses that are being generated at the interface, you can also have to think about this chemical process of turning on and off these Rho signals. Okay.
Can I just ask, Margaret, for a second? On the previous slide, you showed this epithelial network, this tiling effectively. And so it's and it looks like, they're mainly interacting with the E-cadherins. Is there anything happening at the lateral membrane, let's say more sort of extensions of the cell, where they can interact also with, not only with their neighbors, but maybe with distant cells, more distant cells? I think there was some sort of issue with development in terms of bristle formation potentially on the flywing, but I'm not really sure anymore. So
Yeah, so I would think about most of the longer range signaling. I mean, the calcium signaling can propagate pretty quickly, but I think of it as interactions between neighboring cells. There's no physical process that I know of that really interacts long range, although I might be wrong. There are protrusive activities on the basal surface near the collagen matrix.
Yes, that's what I mean, sort of protrusions like.
Yeah, there's protrusions on the bottom side.
Yeah.
So it's interesting for people who study like epithelia mechanics. So there are conditions in which you get dominated by protrusive activity on the basal surface of the cell. You need to have sufficient coupling to the basal surface, but also you need, the cells are generally less packed in those conditions. So for instance, these are the same exact cell types that others have studied where there's lots of neighbor rearrangements and basal protrusions. We've cultured them under conditions that form a more polarized epithelium. And so it turns out just even with the same cell type, how the epithelial tissue is formed can make a big difference of whether you have those or not. Okay, so, well, I'll get into this a little bit later. So we've looked pretty extensively about whether there's effects of this basal interactions. And for what I'm going to be talking about today, it turns out there's very little effect. So we really can focus on the 2D apical surface. Okay, so there's this Rho signaling, which is this chemical signal. Now, instead of being a constant chemical signal, what people have found is that the chemical signal is also temporally structured. So, the Rho signaling is not a static value, say at the junction. It actually increases and decreases over time. So as the Rho signal is increasing and decreasing, you can think about this as a pulse of actin and myosin assembly, again, going down again. And every time there's a pulse of actin myosin assembly, there's a pulse of contraction, of contractile force. And what these pulses lead into, so this is a movie from Tamala Qui's lab, where they visualize this active Rho with an anilin biosensor in GFP. And so you can see these pulse increases of pulse of Rho activity. And if you follow one of these junctions, you can see it shrinking and somewhat expanding, shrinking, somewhat expanding. And what's happening is that this has been called a ratchet of the cell-cell junction, because even though there's these pulsatile nature, those pulses are coupled to junction shortening over time. Now, that's been studied extensively in developing epithelial. It's become clear more recently that there's also pulsatile role just in adherent fibroblasts. So this is more recent work from Perry Nalbon's lab, where if you take a Rho biosensor of just an adherent cell on a surface and you look at it, you can see it kind of flickering. So this temporal structuring of the Rho signaling seems to be ubiquitous in many different types of contexts. And this is inspired more recently several groups to start thinking about the cell cortex as an excitable medium. Now I know physicists usually have, there is a very precise definition of excitable media, but I have some questions about whether this is precisely as defined as one, but the idea that you have a chemical mechanical coupling in the cortex, then that chemomechanical feedback is important, I think is very clear. And I think it's a really, yeah?
One quick question. So you now talk about excitable medium. So how do they communicate for two cells to communicate? Is there exchange of Rho or through a mechanical feedback?
I think that, so in the epithelium, I don't have all these cells, let me make sure, have all been done within a single cell or a large embryo. So a single big cell. And so if you take the context of an epithelial tissue, I speculate that what's happening is if you have Rho signaling and pulses in one cell, that's going to drive distortions, and that's going to induce changes in Rho in the neighboring cell. So I think it is very local. I don't think there's a exchange of Rho, like a propagation of chemical signals that way. I think any kind of stimulation of Rho activity is going to be through mechanical coupling of the interface. That's my thought. Yeah.
Margaret, I also have a quick question. When you said, you know, it's excitable and so on, and maybe it depends what kind of definition people use.
Yeah.
Do you see something? I guess one definition would be that it's sort of stereotypical, right? Like an action potential said, you know, something is happening and then there's a refractory period.
Yeah. So I will speak to that. Yeah, we definitely have evidence of a refractory period. Cool. in the context we're looking at. Yeah.
Thanks.
But it's these papers are really the first. I mean, this one paper from Bill Bement's group in 2015 is really the one that, and the data from that paper is just gorgeous. So that's the one that really uses that term. I have not started using that term yet in our mechanical models, but we definitely have evidence of a refractory period. Okay, so okay, cool. All right, so the problem, okay, so let's just, before I go to the next slide, so you have all these beautiful dynamics, right? The living materials are all characterized by these morphogenetic dynamic processes. The way we have developed thus far ways of characterizing materials doesn't really take into account these dynamics. And so we have lacked tools to be able to structure input signals with the right spatial or temporal structure to be able to probe what the circuitry is, the mechanical circuitry inside living cells. It's just really hard. So oftentimes you can do a measurement and you can watch the dynamic processes going. You can do take a micropipette and yank on something, but it's really hard to probe the structure of the circuit. And optogenetics is really a way to overcome this very generally. And we've used this now 2 times. And both times it's really blown my mind. So optogenetics in the context of sort of cell signaling networks is a way where you express 2 constructs in the cell that are where their interaction is light sensitive. So this is a tool that we've been using that was developed in Michael Glotzer's lab, a colleague here at UChicago. It's called Tulips. So the way this probe works is that light drives the interaction between 2 proteins that are derived from a plant-based photosensitive interaction, where there's a PDZ domain and then this LOVE2 domain with a J alpha helix, and the light basically unfolds this J alpha helix, and there's exposes a peptide at the end that then has a high affinity clamp between the PDZ and this FN3 domain. So what's happening is that when you then conjugate this molecular glue, right, to anything you want. So we put the LOVE peptide onto a constitutive membrane binding transmembrane protein stargazin, and then we bind the PDZ domain to a very high efficiency RhoGEF large. So this is one of these proteins that turns on Rho signaling. So now in the presence of light, what happens is we locally concentrate and recruit spatially this RhoGEF to the plasma membrane, and that should turn on Rho, okay? Oops. So, so this is what the data looks like, we can then shine light on a region of a cell. What you're looking at here is the localization of the RhoGEF. You turn the light on, the protein gets recruited here. We're looking at the RhoGEF directly. You take it off and it goes away. So this is a quantification of the recruitment. You can see the kinetics are quite fast, you know, within 30 seconds or a minute. You turn off the light, the protein dissociates. And I think there are now several different constructs out here. This one, as far as I know so far, is one of the ones that is better at having rapid dynamics. So there are some that are more irreversible or have a very high background level, but this is really nice and reversible. So we can think of this as spatial and temporal control of where you want to perturb Rho signaling. Okay, so Patrick Oakes, a previous postdoc in the lab, had done this in adherent fibroblasts. I'll talk about that maybe a little bit briefly, but what I want to focus on today is the use of doing this in epithelial tissue. And so here, Kate Kavanaugh put these constructs into epithelial cells, made a packed epithelium, and now she can and activate Rho at the cell-cell junction and drive the contraction of the junction. And so you can see the data here. The thick black line is the average of the data. And you can see it's really reproducible. This is data taken from many different cells over different days. You know, you have to calibrate your microscope setup to make sure you're delivering the same amount of intensity, but it's really remarkably reproducible. So you, the cell-cell junctions, we chose these cells because they aren't very mobile beforehand. So when you shine light, you can really see a contraction. And so this data is pretty boring right now, right? you shine light, you make it contract, you take off the light, it goes back to its original shape, right? It's very consistent with the standard Vertex model. It's like a spring. And so there's nothing really that surprising. But then the great thing you can do with this is now change over how much of the junction you're shining light, but also what the intensity of the light is, or how long you're shining the light for. So you can ask a very simple question. If it's really behaving like a spring, you should be able to double the duration over which you've applied the light, and you should get exactly the same results. And the reason I'm building it up like this is because Kate doesn't see the same results. So now if you just increase the duration by which you've activated the Rho, now you see junction contraction, and now you see the initial contraction period, but you see a slower phase remodeling period. And after you turn off the light, now the junction is irreversibly shortened. So now we've engaged the ratchet to be able to permanently shorten the junction. Okay, and then the great thing about optogenetics is now that you can control, you can now experimentally find out what the activation time is, and how much the junctions contract. And so you see this sharp transition to reversible B contraction to engaging the ratchet for the longer durations, okay? And so, you know, again, if you're thinking about morphogenetic processes, usually what you do in development is in any kind of cell culture developmental system is you can make some genetic perturbation and watch how much the contraction rate or shape or how reversible it is, but you can't actually test the kinetics of the process.
Sorry,
okay.
before you go on, just back on that slide. So the bit you shine your light on here, just before you shine on it, it's surprisingly curved.
Yep.
Is there any significance to that? Does that matter in terms
How
of
curved
your
it is
analysis?
at the beginning? No, not really. Yeah, no, because it doesn't. Yeah, you're saying does it matter because the contour length is a little bit longer, or does it matter? Yeah.
I mean, I would have expected mostly straight lines, right? On the basis of your simple.
Oh, the Vertex model. Yeah. I mean, the Vertex model. Yeah, I know this is a complicated. I mean, the junctions are remodeling all the time, right? So they do end up being quite curved at times.
Okay.
Yeah, so that does end up happening, and yeah, we could have a whole conversation about at what length scales you parameterize it as a straight line or not. But yeah, they do, in reality, they do end up being quite curved.
Okay, yeah. Thank you.
And for the purposes of this, the ratchet engagement doesn't depend on how highly curved it was at the beginning. Okay?
Yeah, thanks.
Okay, cool. Okay, so obviously now the Vertex model is broken. All right, you need some adaptation or changes in the length of the junction to be able to get this irreversible shortening. So Sheila Ditcha Banerjee, recently who was at UCL but just moved to Carnegie Mellon, and his student Mike Staden started making modifications to the Vertex model to see what the simplest modifications could be made to allow for this type of adaptation. And I won't go through everything that didn't work, but the result of the model that was sufficient was having two different parameters. One was that there was a constant rate of, there was a rate that, a remodeling rate that depended on the strain. So there's a linear dependence of this remodeling rate as a function of the distortion of the junction. And then the tension in that junction also could remodel, but only did so when it was above a critical strain. So there was a strain threshold remodeling of the junction tension. So if you add these two terms into the into the Vertex model and accounted for this distortion, then you could get a junction that irreversibly shortened. And I would give you a little bit of intuition about what's happening here. So on the bottom curve here, you have, we're plotting junction, this is simulation results, junction strain as a function of time. And so what happens right as you activate the light is there's this acute period of compression over which the tension is remodeled. And then after the tension is remodeled and the length remodeled a little bit, now the strain goes below that critical threshold again. So there's no subsequent threshold reached anymore. So there's no more tension remodeling. So basically all the tension remodeling is happening at this acute period at the beginning of the activation, okay? And so, so this is that was the results of the model. You know, we could then test the parameters, really constrain the parameters put into the model by doing a series of experiments where we characterized the ratchet behavior and tested what I showed you this data before, tested the final rest length as a function of the activation time, but also changed the intensity of the light to know how much light was necessary to engage the ratchet. And really found good agreement between the simulation results and the theory. And with this too, they were able to exclude other modifications to the Vertex model.
So quick question, Margaret.
Yeah. So
in this picture, it doesn't care, it's always about one single junction, so it doesn't care about the other four junctions.
Yeah.
So is it the case? I mean, it looks, looking at the movie, it's almost like it's unzipping. If you are shortening it, then the other two junctions will become longer.
I know, yeah, this is a great question. So the other junctions do not change length. So it's not, so you would expect from a Vertex model that there would be a force, I mean, there should be, that you would expect the other junctions to increase in length, and they don't. So there are other issues with the Vertex model, and we're actually finding this now too, that even what's controlling how much it contracts or the rate of contraction does not depend on this force balance. So there is, I think there's bigger problems with the Vertex model that we haven't addressed yet. Yeah.
Experimentally,
we don't see length. Yeah, we don't see anything length. Yeah.
Okay.
Yeah, I think this is stuff that Kate's is we're trying to understand it now, but it seems that the rate of the contraction is more dominated by the amount of adhesion and how the adhesion sites can remodel than really dominated by the spring, an effective spring constant. I think you know the models that the cable models where everything where the cytoskeletal can be compressible but not extendable, that has a different asymmetric spring constant, I think are a better choice than a completely linear spring where it's the same under compression and extension. But that would be an additional term that we haven't added in yet. Okay, so this is just my big selling point for optogenetic approaches for any of the experimentalists out there. It's just a really great way of testing our pictures of cells as dynamical systems. And I like to do this in the context of mechanics. There's lots of great examples coming out now on also testing gene signaling networks, you know, other kind of chemical signaling networks. It's just a really, really powerful approach for experimental biophysicists. Okay, so now we go to our what I alluded to in terms of the refractory period. So the other thing we can do with the optogenetic approach is then temporally structure our Rho pulses. So this is the example here. We can take one long pulse of Rho, but we can also take the exact same amount of Rho. So the area under these two curves is the same as the area under the left-hand curve, but we can separate those pulses out by a certain amount of time, okay? Before I go any further, I just want to say one thing I forgot to say. One thing you notice with this ratchet behavior when we increase the time is that the length, the amount we could contract the length, really saturated. It's saturated to a really small number. It can only shorten to 20% of its original, or 80% of its original length, which is really not helpful if you're an embryo that wants to completely remodel your junctions. Usually junctions need to remodel 100% of their length. So, so this remodeling by shortening by only 20% is pretty piddly squat. So the question is, how can we overcome this saturation of the shortening? Okay, so so now, so if we change this rest length, remember I told you that there is this, that all the remodeling and the length shortening was occurring during that first stage of this acute turning on the Rho signal. So this is simulation results now. So if we now structure the pulse into two separate pulses, what happens now is you're able to get 2 successive acute remodeling periods that's able to remodel the junction further. So now after these 2 pulses, you're able to shorten the junction down to 70% or 65%. And so Sheila and Mike could then do a full scan of how the amount of contraction shown in the color plot was dependent on the total activation time and the rest time. So this, again, this refractory period. So now if we take one of these activation times, say 45, and increase the rest time, you can see you can go from a period where you only get a final length of about 80–85% of its initial length to one where now you can reduce the length down to 60%. So this is the transition to how do you, the idea that you need this rest time to remodel your length and relax your internal strain to be able to drive another successive phase of shortening. So what that's saying is that this morphogenetic ratchet, right, has a refractory period before it can be active again. And so Kate can go in and do these experiments now, and she can now do 2 shorter pulses of light, and you can see this. All right, so again, we can go back into the experiments and test this idea.
So quick question.
Yeah.
So this comes with the assumption that the 2 cell, well, the junction doesn't go back to its natural length. So if I assume that it's going to adopt a hexagonal lattice type thing, so if your T-rex becomes infinity, I imagine eventually it's going to go back to whatever the typical length is.
Yeah, so we have not seen in the model and in the experimentally, it does not end up, because the tension has been permanently changed in the junction, it doesn't go back to its original length. There is no static rest length, I guess, is another way of saying it.
But if if the actin cortex is remodeling completely, has remodeled completely, then the tension would become the typical tension eventually.
I mean, yeah, I mean, that's another thing. The actin cortex is able to, when it remodels, it maintains it. There's some process of of sustaining that tension, and it doesn't have a stress relaxation time to go back to its original length. I don't know how to answer your question better than that.
So you seem to imply that there's an infinite memory associated to how you put the tension in and then it remembers it.
That's what I think. I don't have anything precise to say about it, but I do think that. I mean, I actually am starting to think about the whole cytoskeleton as having this whole, like, if you think about training it or applying forces to it, that it always, there's a persistence of the memory of what those past signals are or the current signals are because of this feedback of Rho. But I don't have anything more to say. I mean, I would love to know the answers to that. Yeah.
Yeah. Interesting. Thank you.
Okay. So so this has been the physical model of it. And then we can speak a little bit about what the cell biological mechanisms are. So when we look at the contraction, you can do a line scan along the junction, and then all these fiduciary markers come closer together, and there are pockets in which there's a large amount of distortion. It turns out in these large pockets of distortion, you actually see the nucleation of endocytic vesicles there, which then leave the membrane. And so this endocytosis of adhesion receptors is something that's been speculated to be important for adhesion remodeling. What we're seeing is that when you engage this ratchet, that endocytosis is what's is really driving the adhesion remodeling. We're sort of now exploring what endocytic pathway is important there. We did screen a few endocytic inhibitors, including clathrin and dynamin. We found that dynamin was important for the permanent remodeling. So if you inhibit dynamin, now you see the same amount of contraction, but it's completely reversible. So you can't engage, again, this permanent shortening response. So that adhesion remodeling we're seeing is critical for driving that irreversible shortening. Okay.
I mean, Margaret, I mean, basically, doesn't it answer Chufan's question essentially? I mean, you know, once you, of course, remodel the membrane and you make it shorter, is the membrane is only extensible by, I think, 4%. I mean, once it's remodeled, the membrane, you know, as you say, it's irreversible. So I guess it would
yeah,
be.
So you could imagine that, like, yeah, either that changes in length could be permanent from adhesion remodeling. That's true. Or you could think of it as having a steady state tension that's driven by sort of local Rho signaling. And I would say I don't really. Yes, adhesion remodeling is also important. I mean, but you could, there's also mechanisms. I think it's all a dynamic steady state that's controlled, because there's mechanisms to insert new membrane too, right? That are being inhibited. So yeah. Okay. So so the way we're thinking about this is that if you have these pulses of Rho activity, what's happening is that there's a change of the rest length, but also the tension that can be engaged through this initial stage of compression that's driving the remodeling of the adhesion receptors. And over this time, the length is constantly remodeling. And that's really, so all this this ratcheting behavior is really driven by a localized compression-induced endocytosis. So, you know, we've been using this continuum model to describe it, but obviously it breaks down, and it breaks down on the scale of a few micrometers, where you're really thinking about the microscopic picture of adhesion remodeling. And this is something that comes up quite a bit when we're thinking about cell mechanics models, is that the continuum models of cells as sort of active solids or active fluids work sort of shockingly well, in my opinion. I mean, but there's this scale of a few microns where you have these force activated or force sensitive processes that either are, say, driving adhesion remodeling to enable plasticity, or, say, repairing a system to be able to allow it to maintain a homeostatic tension so it doesn't rip itself apart. Okay, so I'll try to be. We've had good questions, so I'll try to make sure I don't go over too much on the second part. So what I want to give you is something that's in press now at PNAS. It's on the archive. It's a mechanism by which this force activated repair can happen. And so here is if you look at an adherent cell and you, say, damage some of these stress fibers, these stress fibers repair themselves so that they don't just allow the whole cytoskeleton to rip itself apart. The way they undergo this self-repair is a process that involves the protein zyxin and its recruitment of other assembly factors that help repair the actin cytoskeleton. So on the bottom is just an image of what the zyxin looks like when you've damaged a whole series of stress fibers, and you can see right after the damage, you see a rapid intensification and recruitment of zyxin.
How do you damage the fiber? Sorry.
This is with laser, like you shine a very bright laser on it. So zyxin, this is the domain structure of zyxin. There's binding to an actin cross-linking protein and an actin assembly factor. And then on the other side of the molecule, there's these 3 limb domains that I'll describe. The zyxin is also important for allowing all the cells to remodel their cytoskeleton in response to stretch. So that's how it was originally discovered. And then this is just quantification of that same movie, showing that basically as you damage a stress fiber and the actin signal is going down in green, you have this very high intensity zyxin recruitment. And the work I'll talk about in this 10 minutes is all done by John Winkelman. So the zyxin, these limb domains, Mary Beckerle's group found that those were what's critical for force sensing. So basically it didn't rely on the alpha-actin and VAS binding. So these limb domains are, they have very poor conservation among different domains, but they have this highly conserved coordination that allows for coordination of zinc ions to form these little fingers. And in the case of zyxin, there's 3 of these limb domains linked in series with each other. And what we're going to call this whole region of the protein the limb domain containing region, or LCR for short. Okay, so it's this whole structure of the protein. So what Mary had found is that it's all those 3 limb domains in series that's really critical.
So just one quick question from the audience, Jane Moore. So the audience said the fibers grow from the center. So you didn't really completely damage the fibers, right? You just
it's not like a complete breakage.
Okay.
And so Sanjay Kumar's done lots of evidence of like laser ablation, and then they just retract. This one is like, yeah, you damage, I don't know, 50% of the bundle.
Okay, great.
Then it's able to repair.
Thank you.
And so, but there's these limb domains, and the LCR has no known binding interaction with actin filaments. So it was not known what they're actually sensing or what makes them force sensitive. So this is from a review from Mary Beckerle's lab. We just had no idea of the mechanism by which they're interacting with the actin cytoskeleton. So that's what John was set out to look for. And he was inspired by this as well, not just beyond zyxin, is because this whole family of limb domain proteins has been found ubiquitously in different types of cell mechanosensing pathways. So these are an example of a proteomic screen for mechanosensitive proteins done by 2 groups almost about 10 years ago. And so these limb domains, so right here at the top is all the class of limb domain proteins, and there's 70 of them in animal cells. And in pink here are all the places of the limb domain structure. So you could find your favorite family class of proteins, paxillin, enigma, ajuba, Tes, like there's just all these different limb domain proteins that are found in different physiological processes. So the question was whether the force sensitive properties of zyxin, one of these, was also found in other limb domain proteins, which we didn't know. So John set up to do a screen of whether different limb domain containing regions of the protein were force sensitive. So this is a screen he set up. He used the stress fiber breakage assay as a way to test whether any of this region of the protein sensed actin stress fiber strain. So what you're seeing here is a movie of full-length zyxin, with a GFP tag on it, so the full-length protein, and then just the LCR of zyxin, so just the limb domains. And then you're seeing 2 different channels, that's tagged with M cherry. Then you're seeing 2 different channels, they both localize to these stress fiber strain sites. And so then if you do a line scan along the stress fiber and look at chymographs of this, you see they both accumulate at this point of stress fiber strain in the upper right. And so then you can take a line scan around 0, right at the stress fiber strain site, and look at the intensity as a function of time, and you see they both increase. And then with appropriate normalization, you can compare the ratio of the accumulation of just the limb domain portion versus the full length portion, and find out how much it's recruited. On the example I showed here, it's just 2 different colors tagged to zyxin, so there shouldn't be any difference, right? So I've shown you a ratio of 1. So he now can then express different LCRs he did it for like 40 different limb domain proteins. And so now these are his results. So the question is, what's the ratio of the M cherry to the GFP? This is the full length zyxin on the left, but then he could find to varying degrees different amounts of recruitment of limb domain containing regions from the zyxin family, paxillin, Tes, and Enigma family, plus a lot of limb domain containing regions that don't recruit at all. So we identified 18 different LCRs that recruit to these stress fiber strain sites. Okay. Now this is a massive family. There's a lot of redundancy in eukaryotic cells, which actually makes it hard to find physiological phenotypes of any one of them. And John came from a background of working in simple model organisms. So in fission yeast, there's only 4 LIM-domain containing proteins. And one of them associates with the contractile ring of actomyosin. It's this protein paxillin-like 1. And so this is the structure of paxillin-like 1. He was able to then take a construct of paxillin-like 1, put it back into eukaryotic cells, and even this LIM-domain containing region sense stress in the actin, in the stress fibers. And it was the only one of fission yeast proteins to do this. So some of these LIM-domain regions are force sensitive, some of them are not. Okay, so what this means is that LIM-domain mechanosensing has been around for a very long time in eukaryotes. And if you look at like a historical plot of like where things emerged, there are constitutive actin binding proteins that have these LIM domains to it that Michael Way studies a lot, that are CRP-like. At some point, he postulates that this mechanosensitive emerges like before fission yeast appeared. But then there was this massive expansion. And when this expansion occurred is right at the development of multicellular life, right? So a time when you think that force sensing might be really important. So we're thinking of these LIM domains as a large family of proteins that really integrate mechanical signals in different ways in the actin cytoskeleton. And so question is, how does this work? And we still don't know all the answers, but I'm gonna tell you what we know so far. Greg Aleutian's lab at Rockefeller has been working on the same problem. He's really interested in the individual LIM domains in one of those zinc fingers. What is the sequence? Are there any sequence specificities of the LIM domains that are force sensitive or not? Okay, and what he found, and this is a paper that is in press at Developmental Cell, so that there's a phenylalanine enriched at site 66. So if you take out that phenylalanine at site 66, you get rid of force sensitivity, but and if you put it back into other constructs that lack it, you can recover force sensitivity of some LIM constructs. Other than that, there's no sequence specificity. When we look at this phenylalanine, what we find in our bigger screen that we did is only 75% of these have this phenylalanine, but there's also 30% of the non-binders also have this phenylalanine, so we don't think it's necessarily necessary, and it's not always sufficient to get this for force sensing. There might be neighboring phenylalanines or tyrosines that are important, but we need to explore that more fully to understand the location. So what John was interested in really was not just a single LIM domain, but really how these LIM domains are packed together to be able to enable force sensing. And so he started looking at the domain organization of the LIM domain containing region. And what he noticed on this plot on the lower left is that if you look at the recruitment of the stress fiber strain sites as a function of LIM domains, all of them had more than 2. So you really need 2 of these LIM domains in serial to be able to get recruitment. And he also found that the ones that bound to that were mechanosensitive had a very short linker. That ones that lacked the mechanosensitivity had a broader distribution of linker sites. So, so he was able to make different constructs with different amounts of serial addition. And if you focus on the cyan colors here, you can see you can take the LIM1 domain of zyxin and just repeat it either 1 time, 2 times, 3 times, 4 times, or 5 times. And you can see the amount of recruitment to the stress fiber strain sites increases. So this really suggests to us that some of these LIM domains, when they're linked in serial with the right appropriately spaced linker, is what's necessary to sense strain on the actin filaments. Okay. Okay. So as I said before, these LIM domain containing region, and how is it working? This LIM domain containing region of zyxin is not a canonical actin binding protein. So it doesn't bind to individual actin filaments in a sedimentation assay. It also does not affect actin polymerization in any way, so it's not affecting actin filament growth or nucleation. And so what we did was we turned to an assay our lab had developed previously to study actomyosin contractility, where you can reconstitute actin and myosin to build mechanically stressed actin filaments and networks. And so here you can build up a network with different types of mechanical stresses. The mechanical stresses in these reconstituted networks can be both compressive and tensile. But they're building up lots of force. So I'll show you two movies. So you then purify the LCR of zyxin in these networks. And now I should just note that this is getting close to single molecule level imaging. This is all done with turf microscopy. And so now what you see happening is that these LCRs really localize to the points of high mechanical stress to the actin networks, and then transiently, and then go away. So if we look at the fraction of events that have a localization, they go to places where the networks are breaking, where these T junctions are about to break. They don't go to T junctions that don't break, and they don't really randomly associate to the network. So they're really sensing places of high mechanical stress, and the amount of fluorescence intensity you think is like about 10 molecules or so. So it's a very low level. So it's a transient localized interaction. We also were able to reconstitute stressed actin networks without myosin 2 using a classical bead motility assay with actin polymerization, and showed there was localization of limb domains to those. So this type of localization of mechanically stressed actin doesn't require myosin 2 specifically. All right, so this is what we know so far, and it requires more careful sort of single molecule biophysical studies, is that we think that places, when there's no force applied to the actin filament, there's no association of these limb domains with 3 serial limbs. But then if you either mechanically compress the actin filaments or tense them, then there's a high amount of association. But it's very transient with just this LCR region by itself. So the way we'd really like to going forward is think about these limb domains, is understand why they get located to different parts of cells, to adherent junctions or focal adhesions or stress fibers, and how they're integrating different types of mechanical stress in the actin cytoskeleton. Okay, so those are what I want to say. So optogenetics really allows us to test these coarse grained models and understand how things are changing with times, and that to be able to have adaptive properties of cells, you need force-sensitive organelles that are allowing for changes in mechanical properties. And to enable that force sensitivity, you need distributed force sensors in the actin cytoskeleton to enable a localized and rapid response. And with that, I'd just like to thank the people in the group who did the work. Kate Kavanaugh did all the work with Epithelial, the morphogenetic ratchet, and John Winkleman was the postdoc who really inspired and drove the whole project on limb domain force sensing. And thank you for your attention.
Wow. Thank you very much. That's really fantastic. Thank you for your great talk. And let's see whether we have any questions from the chat. So we don't have questions from the chat, but whoever's, are there any questions from the audience at this point? You can raise your hand, or you can, Robert. Oh, okay. So by Wooly.
Yeah.
You have a question.
For the
yes.
great talk. Yeah. So when you have this endocytosis, do you see also kind of proteins on the endocytotic vesicles as well?
We see evidence that adherent is endocytosed. Yeah, so that's, we think that we see internalization of adherent.
Okay, cool. Thank you.
Thanks.
Okay. So there's a question from the audience on the chat. So from Ayushi Agrawa, does alteration in mechanical pulsatile motion in the adherent epithelial cells result in cancer cell initiation or play a part in cancer invasiveness? Any thoughts? So
yeah.
maybe.
That's a very big question. I think that, let's see, when there is misregulation of epithelial behavior, that does lead to cells migrating and moving in places in the body where you don't want to go. So I think that maybe, I don't know if there's direct evidence that the pulsatile Rho nature is important, but there's definitely lots of evidence that's showing that shows that misregulation of RhoA GTPase signaling does affect the metastatic programming of cells. And I could work from Val Weaver's lab at UCSF has shown that. I don't think anybody's looked into the temporal structure of the RhoA programming.
Yeah, I maybe following up on that, I guess in terms of the connection to cancer, I guess it's important that a cell leaves epithelia, I guess, becomes essentially motile. I mean, I guess that's one of the issues, right? Yeah. And the next issue is, of course, I mean, Rho signaling is also involved in cell migration itself, right? And,
yeah, so it's Rho signaling is really important for adhesion regulation, and also yeah, Rho signaling is pretty critical for cell migration, especially mesenchymal migration. I mean, there's certain types of cell migration that doesn't require Rho signaling, but yeah, anything where you have adherent cells that are generating contractile tension, Rho is really important.
So you think for cell migration for mesenchymal cells is then basically this ratchet type mechanism you described in the first part?
Oh,
would
yeah,
be
whether
equally relevant. Is it right? Would it be relevant?
Yeah, that's a good question. I mean, I think the I'm not aware of any ratchet process like that. I mean, you could almost think of cell migration as being like an inchworm, like a ratchet between the front and the back of the cell. And I have not seen people, I mean, that really is important for when you establish polarity of the cell, like your front-back polarity. I don't know if the pulsatile nature of Rho is important for that in particular. What I suspect is happening in adherent cells that are very well spread, like a fibroblast, like if you notice in that image of the Rho signaling, the pulses were really sort of diffuse and fast. And I think that's because the whole cortex is under a much higher tension than what you get in the epithelium. I don't know that, and I don't know if that's been measured. But I think the Rho signaling is more localized, and I don't know if I can think of a process where ratcheting like that is important for cell migration.
Mm-hmm. Okay, thank you very much.
I think Alfredo Scottino had his hand raised. Would you still like to ask a question?
Yeah, thank you. I also wrote it in chat, so you can ignore that. Thanks for the nice talk. I was wondering if you have any insight on how the limb domains are able to differentiate between actin filaments under stress or not. Maybe they can snag in.
Yeah, so this is all speculation at this point. We'd like to understand this more. We think, I mean, actin filaments are negatively charged. So we think that maybe between the combination of the phenylalanine, that there's a change, that the change in when you say pull an actin filament, it changes the charge distribution locally. That then through this additive property of the limb domains in conjunction with the phenylalanine senses that charge distribution. And it has to, it's an actin filament, it's a helical structure that repeats. So you could imagine that the same site is exposed at periodic intervals that match up with what you see in the limb domain. This is a lot of hand waving because we have no idea at that scale. We need simulations done, like large scale simulations of the actin filament undergoing mechanical compression and tension.
Thank you.
Yeah.
There was another question in the chat box by Keith. The actomyosin network you made for the last experiment did not contain WASP ARP complexes. Is that correct?
So the actomyosin ones did not. Those were just polymerized, spontaneous polymerization actin filaments. In the ones that did not contain myosin, those did have an actin nucleator in it, like WASP, and an ARP23. Okay, but those were the myosin. Those did not have any myosin 2.
Mm-hmm. Very good.
I have a question. In the last part of your talk, you talk about how the limb domain sends the strain or the stress on the fibers. So in your experiments, it's at a sort of multiple fiber level where we are not talking about single actin filaments. Is that correct? Or you're thinking that it's...
We think the limb domains are sensing individual actin filaments.
Okay.
Yeah, and so Greg Aleutian, he found similar results by doing a different kind of assay, which I think was really elegant. He just looked at actin filaments gliding along a mixture of myosin 5 and myosin 6 motors. So single filaments attached to motor proteins that then could generate places of compression or tension. And he saw evidence of single molecule binding of limb domains to that.
Okay. So
we do think it's a single filament scale process.
Yeah, that's very interesting. So then are you, in your model, you are imagining sort of like a stretching out of this dimers, but is it also possible that is sort of changing the picture of this helical structure and is sensing the change of picture, or you're excluding that picture? So if I stretch out, it's a helical structure. If I stretch it out, the spacing can be further away, or it can open up the pitch. Right? You could change.
Yeah, that's what we're thinking. But we would need simulations, and then so Greg Aleutian is working to try to get a structure of what limb binding to actin filaments. He's trying to actually do the experiment on an EM grid where he can get the structure of what the limb domain looks like on the actin filament. And I think that's the kind of study that are needed to answer that question.
Okay, great. Yes. All
Maybe
right.
one more question, or I have another one. I mean, in the beginning, in the first part, Margaret, you talked about, say contraction or the optogenetics and the shrinking of the cell edge. And it didn't go back to the resting lengths because it was essentially plastic and so on. And of course, I mean, in general, you see these kind of responses, these kind of plastic responses, or I guess viscoelastic, partially elastic but not going fully back. You see this a lot in cell mechanics, right? I mean, I know some other people who tap the cells and then they see, oh, it doesn't come back fully. I mean, it seems like a general problem in cell biology, it sounds like, right?
Yeah, it totally is.
Yeah.
I know it is. Yeah. I mean, so the thing that I don't understand, because I mean, okay, so we know that all these actomyosin arrays are made by different, like, let's just simplify, different amounts of Rho signaling, right? So like, you know, to get an edge with higher tension, you need higher Rho signaling there. To get an edge with lower tension, you need to modulate that. But that means there has to be some feedback to controlling the Rho signaling. And that I don't understand at all. I mean, I think, but you're right, because I do think it's like an actively maintained tension, right? Because, you know, if you disintegrate all your, like depolymerize all your actin, it just becomes a spherical blob, right? So to control your shape, you constantly need to have local feedbacks to control all those tensions. Yeah, and I think how you make it be semi-permanent, right, without to have those feedbacks of the signals is something that I'm really, I think it's a really interesting question, because right now, I mean, like active matter models and like mechanical models of cells are working reasonably well. Like I think they're a good place to, you know, go from, but like we don't have a good framework to understand that adaptation process. And I don't think we have experiments to really test those types of, understand that.
I guess in general it has to do with some remodeling, right? But of course it's very vague.
Yeah, but there's also this sensing, right? Like, it's remodeling, but also has to constantly like assemble the structures to maintain it. Yeah, it's it's a super cool, yeah.
Okay, and in the chat, I think Tom has comments. You want to say,
That's
Tom?
Right. Yeah.
I was just saying, yeah, like if it was just an equilibrium system like the simple Vertex model, then by definition an equilibrium thing won't demonstrate this kind of hysteresis. Because
That's right.
That's right.
So you can get hysteresis in soft materials, right? But like the material needs to be like jammed, right? Kinetically arrested. So yeah, you can explore. Yeah. So one of my colleagues here are trying to explore this in soft matter systems, but like, yeah, I mean, I think cells do that too, right? But they're active as well.
I'd say there's at least two ways to generate it, right? One is to have incredibly slow relaxation dynamics, and one is to be in some miles from equilibrium.
Exactly. So, or like have a constant cue that's telling you like some feedback signal. Yeah. And I mean, I think cells have to have some of the feedback, but I think we could also learn a lot just from the the kinetically arrested systems too, like the soft materials, to understand that. Because so Sid Nagel and then Arvind Murugan here are trying to understand like how do you teach materials to learn and how long the memories last. And yeah, no, I think it's a great question. It's a really interesting area.
Mhm.
So there's a question from Jordan Juritz. Forgive the naive. Okay, you asked.
Oh.
I could read it out
Yeah,
loud.
this is like the bane of every cell mechanics person's question. I mean, like I started in this field 20 years ago, and like actually, I see Andreas Bausch is on the call, or he might have left. There he is.
Mhm.
I mean, yeah, like applying forces to cells is really hard.
Yeah.
So optical traps is too low, right? So they're really like they're a few piconewtons. So, but then also you have to couple the optical trap to whatever part of the cytoskeleton you're interested in. And so magnetic traps can apply larger forces. They're harder to calibrate. You still have this issue of getting the probe into the cell and getting enough access to it. I mean, the challenge of the optogenetic approach is that you never really, I mean, we don't know the forces we're applying, right? We're changing Rho signaling. So it's indirect in some ways, but I think you have enough control over other aspects that it gets you, I personally think it's the best way I've seen to be able to probe local mechanics, because you can really just change which parts of the cell in the same experiment you could really manipulate in different ways. You can couple, so traction force microscopy, you measure the boundary stresses of what the cell is sitting on. And that's a calibrated elastomeric substrate. So you can like measure how much force the cell is exerting on its surface, but it's only probing the boundary stresses.
Okay, thank you. Thanks for a brilliant talk.
Fantastic. Are there any questions? Well, if not, then let us all thank Margaret again for the fantastic talk.
Yeah.
And thank you for sticking around and answer all these questions. It's been great. It's been very interactive. And thank you very much.
Thank you.
Thank you, Margaret.
Thanks,
All right.
Margaret.